Next, I would like to introduce Dr. P.J. Brooks to the stage. He is a program director in our Office of Rare Diseases Research at NCATS. And earlier in his career, he was an NIH intramural investigator studying rare genetic DNA repair diseases. And he is followed by the panelists from his session. Thank you.
Great. Well, thank you, Alice, and it's really a pleasure and an honor to be up here chairing this session. The title is Individualized Therapeutic Approaches in Personalized Medicine, and while that may generally cover what you're going to hear, the reality is that you're going to hear something a lot more specific and I think more exciting, which is the story of the development of an experimental treatment for a little girl with a rare genetic disease and how that was done with collaboration and interaction between the parent of the little girl, an intrepid physician scientist, and interactions with the regulatory agency. And if you've never heard this story before, I think you're certainly in for a treat because it's quite inspiring. But I also think that as you're listening to this, it's worth keeping in mind that this story has much broader implications than this one little girl. And if we think about it right, and if we do the science right as we go forward, it can have implications for thousands of people like her. And we can really get into that later in the discussion in the afternoon. But without further ado, let me get right into the story. So first, introduce Julia Petrillo, perhaps better known as Mila's mom.
So 3 years ago, my then 6-year-old daughter, Mila, was diagnosed with Batten disease, which is a rare neurodegenerative condition. At that time, pretty much nothing was known about her rare form of an already rare disease. There were less than 20 cases in the world, and there were no treatments, there was no cure, and like probably many of you, I was told to go home and watch my daughter lose all her abilities and die in the next 5 years. Before I go on to tell you how my daughter's story took a very unexpected turn over the next year, I think it would be helpful for you to see a little bit, get to know me a little bit. So I would like you just to see a very short clip of a video that I made right after she was diagnosed that would help me race for her life. So I'll continue the story after.
So our daughter's name is Mila Virginia Makovets. She was born on November 5th, 2010. When I think about Mila as an infant, I think about smiles and laughter and health. And
half an hour after she was born, she was strong enough to hold on to my finger and I could almost pick her up off the bed.
When Mila was a toddler, she was a climber, she was a hiker, she was a runner.
How old are you, Mila? Here's our sugar high daughter.
Slide, slide, slide.
Very physical, always loved to ride a strider.
Nice, Bug.
Play in the snow, go sledding.
Coming!
We noticed that Mila was getting stuck on her words. She would say, "Mommy, Mommy, I have to. Mommy, I have to," and not be able to continue. And then suddenly, around 3 years old, we started noticing that her feet were slightly unturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way, and then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. You know, we took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She could no longer see anything.
When Mila was diagnosed, I have to be honest, the first reaction I had was relief, and it felt very guilty to have that feeling. I had gone to 100 or over 100 appointments with doctors and therapists around the country, dragging my infant son with me at the time as well, and no answers, like many of you. And obviously, it took me a very short time to read about Batten disease and realize that my daughter had been diagnosed with one of the worst diseases. The way I would describe it would be a combination of epilepsy, blindness, dementia, Parkinson's, all combined in a little 6-year-old girl who had previously been very advanced and outgoing. And I was being told that she was going to lose all of those abilities in front of my eyes in the next few years and then die. I looked over at my daughter when she was diagnosed, and I saw her smiling and laughing despite having just lost her vision. And I thought, if she's fighting, I'm going to fight and do everything I possibly can, even though I'm being told that we're at least 10 years away from finding any cure, any treatment for this disease. And I started doing what probably many of you have done, where I started reading everything I could get my hands on, speaking with scientists and doctors, speaking with other families that had started foundations, and telling Mila's story to anyone and everyone that would listen, in hopes of raising enough money and catching the attention of scientists that would help fight for her life alongside me. I started Mila's Miracle Foundation. I somehow raised quite a bit of money, and people started following Mila's story, and 7,000 kind strangers in 55 countries really wanted to know what was happening with her, because she was losing her abilities very quickly, and I was fighting for her life for a gene therapy at the time. However, there was a problem. Mila's diagnosis was only half diagnosis. It's an autosomal recessive disease, so she has to have one mutation from myself and one from her father, and all labs could only find one. And the problem was that I had a 2-year-old son, and my daughter, as you saw from the film, was very advanced and normal at 2, and so was he. And there was no way to test him unless I had a full diagnosis. And there was no way for me to be totally sure that I was going after the correct gene for gene therapy unless I had a true diagnosis. So I pushed through social media and with a plea to have any lab out there that offered whole genome sequencing, of which there were a handful, not even at the time, to help me dive into this and try to figure out where this missing mutation was. I'm going to let Dr. Yu tell a little bit more about the details of what happened after that point. But during that 1 year, I crossed paths with Dr. Yu through social media. And what started as his incredibly kind offer to help me find that mutation, and eventually he did. And eventually he was able to tell me that my son was free of Batten disease and I would not have motherhood ripped from my arms. It turned into a pretty amazing story. And 1 year later, after Mila was diagnosed, we moved from Colorado to Boston, and Mila received the first dose of her drug that was called Milasin after her, and she became the first person in the world to receive a drug tailored just to one person. And later in our discussion, we will talk more about what the implications of this could be, but at some point I will say that I realized during that year that Mila was being given this really incredible chance, and she was losing her abilities by the month. And I didn't know if it was in time for her, and I still don't know if it's in time for her. But I had so many parents reach out to me from so many different diseases all over the world, some of them with emails that really hit me hard, telling me that they had just found out that their second little child had just been diagnosed with the same disease that their 5 or 6-year-old was dying from. And it just really hit me that this was more than Mila, it was more than my daughter, and it was just a huge opportunity in a world, this rare disease world, where there are no tools in the toolbox. And it started hitting me that this might be a tool that could potentially help many, many children across many diseases, not just Batten disease. And over the last few years, this has been a story about Mila breaking the ice, but it's really turned into a story about something much bigger, and something that Dr. Yu and I have worked really hard on together with everyone at the NIH and the FDA and many other organizations to try to open up a new path that could give other children like Mila a second chance at life, hopefully, that are diagnosed even earlier before their symptoms take over. So thank you for letting us tell our story, and I'll let Dr. Yu come up and finish the story right now.
Okay, now I'd like to introduce Dr. Timothy Yu from Boston Children's Hospital, who can tell you about his efforts working with Mila to develop Milasen.
Thank you very much, PJ, and thank you, Julia, for introducing this story to the audience. First, it's a great honor to be here in celebration of Rare Disease Day, working together with the NIH and NCATS, very active collaborators, and the FDA being on the stage with us is only appropriate because this project has been a very collaborative effort across multiple parties. I will share with you the details of what happened after the point where Julia left off. By way of introduction, I'm a neurologist at Boston Children's Hospital and Harvard Medical School. I run a laboratory there that for several years had focused on the diagnosis and discovery of disease genes, but had not yet made serious inroads or put serious thoughts together into treatments. But as we discussed this morning, we are getting better at finding the names of the genes and the diseases that ail us. And this case that we're discussing today points towards exciting new opportunities in turning those diagnoses into potential interventions that someday may help many, many patients. Now, Julia has shared with you the story of her daughter, Mila. As you heard, she was a 6-year-old at the time when we met her. She had a terrible diagnosis. Her physicians had diagnosed her by skin biopsy, and she had been shown to have a condition called Batten disease, which, as Julia pointed out, is an exceedingly rare disease that's progressive, fatal, ultra rare, meaning that in 2016 and 2017, when the diagnosis was fresh, there was no drug company interest in her particular subtype of disease. Now, there was a glitch. As Julia shared with us, her expert clinical team had made the right diagnosis, but molecularly, it was only half of a diagnosis. This is a disease that requires 2 mutations, and 1 mutation was evident on clinical sequencing, but the second was nowhere to be found. And that highlights an issue that was discussed in the Diagnostic Odyssey panel earlier this morning, whereby 99% of the genome is dark matter. It's not yet included in most clinical sequencing available today. Certainly not in 2017, but not included, it's still in many diagnostic labs today. And the only way to interrogate this dark matter is to do whole genome sequencing. Well, that's where a stroke of fortune connected Julia and ourselves. We as a laboratory had been doing whole genome sequencing for about 7 years prior to meeting her. And I happened one day across this post on Facebook, forwarded to me through my wife, through a series of colleagues that were in the Physician Mothers Group, a Facebook group that is very active in the Massachusetts area and a useful networking resource. I learned there of the Petrillo family and Mila's plight, and the fact that she and her clinicians were looking for someone who could provide whole genome sequencing to answer a very critical question. And so we reached out to help, and in March of that year, we're able to offer her an answer. We found something rather unexpected. We found a misplaced segment of genetic code that was sitting right here in the part of a gene that was not normally interrogated by most companies. And the consequence of this misplaced segment of code was that it caused a splicing alteration, a change in the assembly of the gene, and it essentially interrupted the instruction set of this critical gene. Now, March 2017, we were thinking about this, and we had a flash of insight, which was this: what if we could devise a way to actually silence this? Now, where did this come from? This concept actually came to us from a drug approval that many of you know, that's been talked about here, I'm sure, in several recent years, whereby a drug called Spinraza for spinal muscular atrophy is now FDA approved and is delivering wonderful benefits to children with a rare and fatal neurodegenerative condition called spinal muscular atrophy. This type of drug, Spinraza, is in a class called an antisense oligonucleotide. And it's a string of 18 letters, chemically synthesized, but designed to target and home in to one specific spot on the genome, where, what does it do? It finds the right target gene and it restores normal splicing. It changes the way the gene is spliced. Well, that led us to think, could we replicate this same development path except for our patient? And so beginning in April, we designed a plan and began implementing a plan to try and design an individualized medicine following the Spinraza template, whereby we could take this abnormal splice event, test for it in skin cells isolated from our patient, and then design a series of antisense oligonucleotide drugs that might have a chance at reversing this. That set us down a path that by July of 2017, we had initiated screening in patient cells and we were beginning to see positive results. And that a few months later, we were able to show that a drug made using this strategy could restore proper gene splicing, and the details of this are not important, other than to say that this is the amount of normal splicing that was going on without treatment, in red, 7 to 11%, and that with treatment, we were able to triple or quadruple that. And a few months later, we were able, with the help of collaborators at Northwestern, show that these drugs were not only improving the RNA message, the gene mRNA message, but they were actually making the cells healthier. This particular gene is required for proper recycling of cell wastes, and in the absence of this gene working, Milas cells were accumulating these white bubbles that represented proteins and lipids, fats that her cells could not get rid of. However, treatment of her cells with our drug reduced the number and the size of those down to really nearly looking like normal. Throughout this time, we were following Mila's progress closely, and we were engaging with the FDA because this was, on one hand, a scientific experiment, but on the other hand, this was a compassionate use clinical attempt. And we engaged with the FDA, in particular the Division of Gastroenterology and Inborn Errors, and we said, we've got our sights on something unusual here. We have a drug that's working well at the bench. We have a young girl in dire clinical need. Can we come up with a path together to implement this and allow us to proceed with administering this agent to our drug? And they cooperated and collaborated with us, and together we were able to solve many unusual problems that had not yet arisen in FDA guidances. There's no existing guidance for cases as small as one. For instance, making the amount of this drug, making this type of drug quickly and safely is a really critical step. But typically, to get a drug like this manufactured takes 6 to 9 months. It costs many, many hundreds of thousands of dollars and generates usually quantities that would be sufficient for 50 times the number of patients that we were talking about, one. But with a lot of creativity and flexibility on the part of the FDA, as well as the help from many industry collaborators, we were able to devise ways to address these issues and do this efficiently and safely. In 3 months' time, we were able to manufacture the drug, start critical toxicology studies in animals, and then launch the trial, which began on January 31st, 2018. And this work has been published and described in the popular media quite widely. What did we see in 2 years now into this trial? Most importantly, this effort has appeared to be safe and well tolerated with no adverse effects to date. And is it working? Well, this is not a fairy tale. We can't say that we haven't restored beautiful young Mila to her previous state. But we have strong evidence that this drug is helping. It's improving her seizures, and we believe that it is slowing her course of disease. This plot simply shows the seizure response to our drug begun to be administered at the end of January 2018, and you see that seizure counts and seizure intensities dropped significantly over this time period. This work has received a lot of attention in the news, most notably for the fact that we went from patient referral to first dose in just 1 year's time. And it has a lot of implications. It's a personalized medicine. And it was a huge effort to develop arguably one of the very first examples of a truly individualized genomic medicine. It required intense collaboration between academia, patient foundations, industry, CROs, and regulatory agencies. And we've been discussing this with many stakeholders because this is a drug for Mila, but it also has implications much broader beyond that. And just a few of the points that I'd like to highlight. It raises the prospects that we may be able to think of paths by which we can develop treatments that are targeted at the class of genetic mutation that a patient has, and not necessarily only by the particular disease that he or she possesses. It allows us to begin to have visions of genetic therapy clinics that might be able to turn around these types of approaches quickly if we can navigate the next few years successfully and carefully. And it also raises challenges that we confronted in this case, but will require further work to get just right. How does one manage safety? How does one scale this approach? How do we ensure that this type of approach can be accessible so that patients aren't having to, in addition to coping with their conditions, also raise money to support these efforts for their children? So I look forward to the discussion that will follow with our panelists. I'll end with this slide simply to impress upon you that this is work that requires many, many different teams from many different disciplines, across agency, academia, and industry, and patient advocacy groups. And we look forward to exploring this with them in the years ahead.
Okay, and last but certainly not least, I'm happy to introduce Dr. Petrillo Sampuco from the FDA CDER, who was involved in the regulatory aspect of this effort, and to talk about her perspectives.
Good morning or good afternoon. I'm Petrillo Sampuco. I'm a clinical team leader in the Division of GI and Born Errors Products in the Office of New Drugs at CDER in FDA. By way of background, I'm a pediatric clinical geneticist, and I practiced clinical genetics and did clinical research prior to joining the agency. I was fortunate to be a part of the team who was involved, along with Dr. Yu, in really kind of bringing this treatment to Mila. So I'm going to give you just a little bit of a flavor of the regulatory perspective and kind of put that piece of the puzzle together. This story is clearly very unique, very fascinating in its own way. I would say it's really a prime example of really things falling into place very rapidly and efficiently. And I think it's because of the very close collaboration of all the different teams and folks that really needed to be involved. From the agency's perspective, of course, there was a lot of work done prior to us being involved, but our team's approach really exemplifies, I would say, this concept of regulatory flexibility. So I know we all talk about regulatory flexibility as something new, but it really is not. it's something that, certainly our division and many of the review divisions dealing with rare diseases have been practicing or using for a long time. The regulatory flexibility is widely recognized within the regulations, and of course, one area that it is truly needed and applied are the rare diseases. So in this particular case, our team did a really extraordinary work and put together this very collaborative effort to really try to think creatively and think flexibly about what really would be minimally required to really get this treatment to the patient as safely as possible based on what we had at the time, but also being quite collaborative with the physician, try to really be in touch very frequently with updates and emerging data to really create this very novel path. So, clearly this is a very new area, really an uncharted territory for both the science and the regulatory piece. So this is one of a just handful of cases, and there's some others also in the agency where there is really a very targeted therapy for a single patient or really a handful of patients. In this case, it's really been rare neurodegenerative diseases which are serious, obviously, and life-threatening. So the considerations in that aspect are very different than how we typically think of applying our regulations for drug approval or drug review. In this case, this unique approach that we need to really use for review of those applications is something that is quite fluid and dynamic. I think we all continuously are learning both from the scientific perspective, of course the patient family perspective, and the agency's perspective. So we have had plenty of discussions with Dr. Yu and his team, kind of putting our heads together and trying to figure out, how best to really provide this promising treatment to Mila, but also making sure that we minimize, to the extent possible, potential safety concerns. In terms of our approach now, this is, of course, evolving. I would say that the single cases, and one of those being this treatment, have been addressed really individualized, just as the treatment is individualized. So it's very unique in the way that the type of disease, the progression of disease, the drug type, but also the class of the drug, in this case oligonucleotide, but also the toxicology data that's available or may be needed to proceed, are all taken into consideration. But also, of course, recognizing that we couldn't possibly get what we typically would ask for, like in a drug development program. I would say so far the approach has been very nuanced, but also very individualized depending on the context. Of course, coming into this era, as previously mentioned, of this therapeutic approach being more widely available due to advances in technology, but also in diagnosis in genetic diseases, we will be seeing more and more of this, which is really exciting, but also, of course, very complicated. There's, I would say, ongoing efforts within the agency to really identify some basic principles or a basic framework to both provide some guidance and some transparency and a kind of a structured approach to this novel paradigm. This, of course, will be very dynamic because this is a new area and everybody's still learning. And so what I anticipate will happen is that we'll start with basic structure and basic requirements, which of course will be probably different for each application depending on the context. But then as emerging information exists, and of course with input from patients, families, researchers, I hope that this will really evolve over time to the extent that kind of the upfront work that was done in this case may be much more streamlined and efficient. So I will end there. I want to say really thank you for this invitation and the opportunity to speak here. I hope that, from the regulatory perspective, we can really be more involved in these collaborations because I think they're truly important for the patients that we all serve. And so I really look forward to the discussion and having any questions. Thank you.
Thank you, Petrillo. And I think what we'll do now is I've got a couple of questions for the panelists up here that we'll go through, and then towards the end we'll open up for the larger audience. And I think I'll start with Julia. You mentioned that you get a lot of queries from a lot of other parents asking you about this process. And I wonder if you could expand a little bit on that and also try to talk about maybe some of the misconceptions that people have that you've helped them to address, because I think that might be relevant to some of the people in the audience here.
Yeah, that's a good question. Milas Story has been, in the press now quite a lot, in about 50 or so countries, I think, in the last few months. And even over the course of the last 3 years, as I was fighting for Mila, I've had parents across probably at least 100 diseases reach out to me on a regular basis. And one of the most common comments is, we're racing for a gene therapy. We all know that gene therapy requires understanding the gene if it is monogenic, and is kind of the only tool in the toolbox for most of us right now. And the question I get is, well, it seems that an antisense oligonucleotide, or ASO, like Mila's treatment, can we do that too? How do we find someone to do that? And how much money does it cost? And I think that while this is really exciting, and I want to start with that because this is a potential new tool in the toolbox, and it's going to be, together with the FDA and the NIH and many others, explored very aggressively and safely in the next few years. It is the beginning. Mila has been treated, and there's this great hope, but we need to face many of the important issues and concerns and work through each one of them, which we are all doing collaboratively right now. But it isn't necessarily for everyone. There's an estimated maybe 10% of mutations for monogenic diseases could be hypothetically amenable. And while that is just 10%, we all know that just 10% could add up to quite a lot of people. So it isn't necessarily as simple as just saying, let's go after an ASO. A patient, and I'll let Dr. Yu explain that more in a minute, but that the patient has to really fit the criteria, and there have to be a number of tests that are done in order to really feel that that child could benefit from an ASO like Mila's. And so I think that it isn't just a question of, do we go after gene therapy or do we go after an ASO, which is the first question I get from most people. And another question along those lines is, first of all, where do we go? And right now, Dr. Yu and I are working on kind of coming up with a framework for where to send people. Right now, I certainly am always open for anyone to email me, and I work closely with Dr. Yu, and he has genetic counselors that can handle that in a more kind of careful way to assess children. It's not myself, but I'm happy to take any requests and forward them on to Dr. Yu and his team to assess. But obviously that's not scalable in this minute in time. I like to be able to tell parents somewhere to go and not leave them hanging. I also think that finding more Dr. Yu's is something that we're all working on in terms of building a network of more physicians and PIs that can take on treatments like this. And all I can tell you is that we're working really aggressively and carefully to try to build that network so that there is a ecosystem for many more treatments like Milas. So I hope that that's helpful, and I don't know if, PJ, there's any other kind of things that you think would be helpful to address.
No, I think that's good. Like I said, we should get some questions for the audience. So let me then go to Petrillo and to think about if you could expand a little bit on the regulatory pathway that was used here, which was expanded access, and how that applies to this single patient oligonucleotide, but also how that pathway is different from some of the other pathways that we might think about in early trials in rare diseases, like first in human, like a standard clinical trial, or compassionate use. If you could kind of talk about that.
Sure. So, typically, what we see is somewhat of three different types, right, in the regulatory realm. So one would be your typical commercial IND or investigational drug application, where, of course, a sponsor, which is typically a company, wants to start trials, right, and in a disease. Then there's, of course, academic physicians doing research, human research. And then the third path that's really reserved for serious and life-threatening diseases, and, of course, rare diseases, many of those are, is the expanded access route. So the expanded access regulations, really what that means is that the goal of this pathway is truly for treatment. So the goal is that if there's one patient or a few patients who may not necessarily qualify for a clinical trial that's ongoing, or maybe there is no clinical trial ongoing for a particular disease, then there is this opportunity to get treatment with a drug through an investigational drug application, specifically for treatment use. So this type of applications, like the one that we received for Milas, have a little bit of different considerations than your, I would say, garden variety IND for a commercial trial. One important piece of that is that there has to be some prospect that there's going to be benefit to the patient. And so since the fundamental, tenet of having expanded access to provide treatment to patients, then there should be some expectation that the drug will have a pharmacologic activity. And in that sense, there's a lot of thought put into what kind of data does somebody have to really substantiate or justify that, yes, this drug is really not research, right? It's more we have a good idea of what we expect to happen or anticipate. And then, we hope to really offer that to the patient. So that's one important piece. And then, the second piece comes in where you have children involved, right, which is a very different kind of ethical considerations when you involve children in research or even in expanded access use. And so a lot of this framework of having a prospect of clinical benefit comes into play. And so we oftentimes ask about potential, non-clinical animal data, in vitro data, to really show that there's some expectation of efficacy. In this particular case, there was actually a very good data from the patient themselves showing really dramatic improvements, in the cells tested and in the expression of the gene and others, which made us much more comfortable saying there is an anticipated effect, at least in the molecular level. So in terms of the question of, how do you fit it in the kind of regulatory paradigm, so it's really hard to fit it. I think this is a very novel way. I would say that overall in rare diseases, this traditional paradigm of phase 1, 2, 3 trials is really, comes into question of how you can really apply that. We see that over and over, that there's this fluid, drug development in rare diseases, and that's for various reasons. Of course, there's not a lot of patients. There's very limited natural history information. And so, we try to come up with creative approaches of really try to get the treatments out there, of course, with enough information for safety and efficacy, but also in the most expedient way. And so the lines, between phase 1, phase 2, phase 3 are really very blurred. at this point in rare diseases. Of course, when you have kind of this novel therapeutic approach of having this individualized therapy, you have one patient or just a few patients. That's really no longer a trial, right? So depending on which way you go, either a research protocol with many patients or a single IND for a single patient, like in this case, the considerations are very also individualized. So I would say this traditional framework of clinical trials is really out of the window in this case. And so that's where this flexibility or creativity and collaboration comes in. I think at least in our division and many others who deal with rare diseases at FDA, there's really a wide recognition that we're really not dealing with your kind of this bread and butter, right, clinical trial development. This is just revolutionized, and this is very, very different, and that requires us and everybody else to think differently as well.
Yeah, I was going to say, I'm sure people really appreciate this flexibility from the FDA, and I think we just heard it. And so going from that, Tim, if we can talk about the idea of how this might be expanded, you know, something you and I have talked about, that in a sense, Mila, in one way to look at Mila's disease is that she has abnormal splicing disease, like many other people would have, and some of those would be actionable with an antisense oligonucleotide, and how you've kind of thought about expanding this more broadly.
Great question. So I think, PJ, kudos to you and Anne and Chris for really having, at NCATS, for having highlighted the challenges that the rare disease community faces. There are so many diseases, and how do we go after them one at a time? Are there new creative methods that could be applied to make the process of developing treatments more efficient? And I think that is one of the key opportunities here, that what we had here was alignment of many things lined up to allow us to help you, Julia, and your family. But one generalizable piece was this, that your daughter has a mutation that has a very defined molecular mechanism, and we have brand new tools in the toolbox that we have experience in thousands of patients that are really good at fixing those defects. The splice defect that your daughter had was something that we have tools that can to address. And so if we can shift to bucketing our approaches and bucketing our treatment strategies by mechanism, as you all have done a great job advocating for, then that is one of the ways in which this experience can generalize to many other patients. So, and I think that that's a very real possibility. Now, I know that this case and the prospect of expanding this out to many folks offers a lot of hope, and I think that that's why it's attracted the attention that it's attracted. I do have to appropriately and responsibly caution that this has been done once, and we have published only one paper on this, and it's going to take quite a lot of work to really take advantage of all that opportunity. Scientifically, we need more experience, and we need to see how more patients respond to this type of approach. And then systemically, there are lots of hurdles that we could overcome in this first instance that will be harder to overcome once there are 10 patients, 100 patients, 200 patients. It takes a change in attitude, but also a change in infrastructure to deliver this kind of care. But I think that if we do it right and we play our cards right and explore it carefully and responsibly, then maybe we can bring that about. It'll take time, but maybe we can bring that about.
Great, thank you. Maybe what I'll do now is open up if anybody in the audience has any questions. Oh, up there?
Can you hear me?
Yes.
Great. So first of all, incredible story. Thank you so much for sharing with us. Julia, you mentioned 10% of mutations can work with ASOs, and I assume that's the percent of mutations for which silencing that part of the gene or skipping that part of the gene would help to fix the problem. In the spirit of increasing this percent, I noticed there's some research papers showing that you can increase defective gene or protein expression by using ASOs on upstream open reading frames, essentially silencing the part of the gene that modulates or suppresses gene expression. Are people looking at this? Is this a valid way to increase that percent? And is this potential use for another use case for this?
You're exactly right. So we estimate, and I should emphasize, these are back of the envelope estimations. So but around 1 in 10 individuals with recessive diseases may have a mutation that alters splicing in a way similar to Mila's. But there are additional tricks whereby ASOs can work. Now, those tricks haven't been fire-proven in the way that splice modulating ASOs have been fire-proven in Spinraza and a few other examples. But it's possible that this could generalize beyond that.
I think what you said about changing the terminology to gene splicing therapy and then being able to bring many more rare diseases under that umbrella is brilliant, and that's a great way to go. So my wife has Crohn's and myasthenia gravis. And when she was in crisis from myasthenia a year and a half ago, she couldn't get Rituxan because the insurance company said, no, we don't think so. Even though the FDA had broadly stated that it was good for certain diseases, and there are a few others, we're not opposed to those either. And the doctors wanted it, the hospital wanted it, and the patients obviously wanted it, but the insurance company said no. And so we had to fight with peer reviews to try and get that. So for our, especially I guess our FDA representative, what can we do to get existing drugs that have already been developed over years and years and billions of dollars, what can we do to get them more readily available to the patients so that the insurance companies can't just say, no, we don't think so?
Thanks for your question. Can you hear me? Yeah, so we get that question a lot in terms of insurance coverage, and I don't think we're the right people to answer that. But at least from approval or regulatory perspective, we do see cases that where approved drugs or off-patent drugs are studied in rare diseases for a particular mechanism of action and for novel uses. So what I would say is the first step of using a drug safely, of course, and effectively is to actually study it in the particular disease. So if it is a drug that's already approved in a common disease or a different disease, it's often, of course, really useful to study it in a particular rare disease, provide data to support that it's actually safe in that patient population, and also assess its efficacy in that patient population. And the same goes for rare diseases. And so, that really, creating a label for the particular disease and the drug, I would say is the first step to really getting the information that many stakeholders need. That, of course, is the physician who's going to prescribe it, because if it is an off-label use of a drug that's approved, do you really know about what safety to expect? Do you know what to monitor for? And also, do you know whether it's going to be effective and in what aspect of the disease? And of course, the patient needs to have that information to the extent possible. And by extension, I would assume that the insurance companies would be looking at that to kind of substantiate and justify their decision. So, I can't really speak to the kind of insurance and our reimbursement perspective, because obviously that's really not our purview. But I think from a regulatory perspective, studying a drug in a rare disease, providing information on safety, efficacy, and labeling it appropriately, I think it's a very important first step in that direction.
The excuse that the insurance company gives is the FDA didn't specifically say it's okay for this disease. So the more that we can get FDA to quickly go through a broader spectrum where it can be applied, the better the patients would be.
Yeah, no, that's right. And so I would say that the study of those drugs is not really what we do, right? And so I think there has to be some study in terms of clinical trial or otherwise from a drug developer. And of course, where the agency comes in is when we actually interact with companies or we actually get information or data to review to support some claims. So in terms of whether the agency would be able to really promote that, I think it's a little bit of too optimistic or a little bit not realistic, because this is really not what the agency's work is. I would say that this is an important piece of labeling new drugs for rare diseases in particular, but I think it's a matter of really the community coming together, drug developers, and of course FDA is always happy to be involved for guidance and collaboration to really label those drugs appropriately. But thank you for your question.
Thanks. I'd like to kind of move on because I think you got a couple more people now.
Yeah, hi. I just had a question for Dr. Yu. Do you see a path forward somehow, whether it's something similar to this, for autosomal dominant neurological disorders?
I would say that absolutely yes, it's entirely possible. So there are different challenges with autosomal dominant diseases, but some fraction of autosomal dominant diseases, a small fraction, but some measurable fraction, is due to mutations like this. And if one can deploy an ASO to restore function of that defective copy because it was a particular type of splice defect, then yeah, I would say that there's a shot. There are other applications of ASOs that are being utilized for autosomal dominant diseases that don't require one specific type of mutation. There's some really fascinating work in Huntington's and ALS that's coming out, that has come out in the last few months and is ongoing. So I think that there are going to be multiple opportunities here, and each one will bring on a little bit of a different risk-benefit profile. Some may impact one patient, some may impact hundreds or thousands of patients. So it's not all going to be exactly like the Milas story rewound, but there's lots of interesting ways that this is going to continue to go.
And, is there a special issue of dealing with the brain versus other parts of the body? Is there, as far as drug delivery?
So I'll say that from our standpoint, the reason that we decided that it was reasonable for us to offer this hope to Julia's family was that we were impressed that the safety profile of this class of drug given in the brain was particularly favorable. This would have been more challenging if we were trying to give it IV or target something systemically, and in my lab's estimation, that's not something that we're ready for to do at this scale yet. But for central nervous system diseases, I think that these are cases that we are considering.
How did you deliver the drug then?
It's actually given by spinal tap. Okay. So a spinal tap given once every few months will distribute the drug throughout the cord and the brain.
Yeah, and I would add, I think that brings up an important point about these neuronopathic rare diseases, right? And so what that means is really a rare disease that has neurologic symptoms, and especially ones that are actually progressive over time. So this area hasn't really been, explored very much. We do see that some of the routes of administration that can get, of course, closer and closer to the brain have been tried. So one is intrathecal, another is intracerebroventricular, and so there is a drug approved that's Spinraza for CLN2 disease, another type of Batten disease, where there's actually administration in the brain through a port. Those are, of course, very novel and risky approaches, interventions. But of course, the bottom line being, how do you get the drugs into the brain? And that's hard just because there's this blood-brain barrier right in the middle, and and there's reasons for having that. But of course, that makes it challenging. I think that's an area that's being developed and looked at from many developers, and it's actually very exciting of looking at novel approaches of actually administering those drugs within the central nervous system. It is, at this point, I would say there's limited, experience with that, but certainly some specific routes of administration, like the intrathecal route, of course, are more viable than giving it intravenously.
Thank you so much for giving me the chance to ask the question. This question is for Petrillo. As far as I know, for FDA, you have applications based on diseases. But as we have seen today, and also in rare disease arena, we will have less and less patients, and we won't be able to do clinical trial phase 1, phase 2, phase 3, and they will have to be more personalized. And is it possible to focus on mechanisms rather than diseases? For example, the mechanism that you have found about splice variation, right, or correcting the splice defect, that is actually seen in many other diseases, not only in that disease, right? And if that application is, one route, or for example, mitochondrial dysfunction, or, axon transport defects, or astrogliosis, and these are seen in some patients, and rare disease patients, but in some of them, because there's a big heterogeneity among Alzheimer's or Parkinson's or ALS patients. And if we focus our attention to the mechanism and develop drugs for the mechanism, that would also solve the question of the other gentleman's question. Is that possible? Do you think we can go towards that direction in the future or in the near future?
Yeah, that's a good question. So the example that I can give you, and that's actually currently being done, is in oncology. So there's been an evolution of this paradigm over the past decade or more, where initially, there were drugs specifically targeting a tissue or an organ, and now it's very much more this tissue agnostic approach. I'm not an oncologist, so I can't really talk about this, but at least that approach is widely used nowadays, and it's quite revolutionary. So, do I see that happening in rare diseases? Yes, of course. The mechanisms many times overlap in rare diseases. I would say that we do see clinical trials done in categories of diseases or groups of diseases, just in an effort to increase the number of patients, obviously, for clinical trials. There's many different ways to do that. Some creative ways to do that. One example may be combining population of patients in one trial, but then analyzing patients differently. So it could be potentially, just theoretically speaking, that you have a particular category of diseases that have a similar or the same mechanism. Then you can include patients who may potentially have different symptoms or different manifestations or severities in the same trial. So having kind of more liberal inclusion criteria, but then you have to really try to make sure that you compare the right population to the right population. So apples to apples, there are comparisons. There's ways to do that statistically and clinically, of course, but you may be able to have ways of establishing different efficacy populations, for example, and analyze them separately. And that's just one way of doing that. There's probably many others, and I think it's a matter of kind of people coming together and seeing how can we tackle this. But I think, just to answer your question, I think it is very possible, and it's in the horizon. And especially since we understand more and more about the actual mechanism of the diseases. And so, that actually brings up the point of really characterizing this natural history on the molecular level and how important the basic science is to really get to that point.
Thank you so much.
Yeah, that question might come up again in the discussion this afternoon, and I've got some thoughts on that, but I'll save them for this afternoon, so if you want to stick around. But I think it's a great idea, and we at NCATS are very excited about moving that forward. I have a couple more minutes. So, are there any more questions in the audience? Yes.
Hi, thank you so much. This question is for Julia. Hi, Julia. I'm also from Denver. I've followed your story very closely. I am a parent of 2 children who had mitochondrial disease, and they both passed. I think for our community that is watching you navigate this road, and knowing that there are many variables that had to happen for you to get to this place, what what do you say to parents who are watching their children die, for lack of a better word?
It's a good question. It's a hard one to answer. I do realize that in Mila's case, a lot of stars aligned for her, a lot. One of them was that Mila really caught the attention of a lot of people in science and outside. Dr. Yu put together a team of hundreds of people. The FDA was incredibly receptive to having a discussion that required a very different mindset than they were used to. I mean, so many things aligned. Spinraza was approved right before Mila was diagnosed, almost the same week. And I believe that there are many, many parents out there fighting, and there's a number of us who are fighting beyond our own children. I really, truly don't know if my daughter's treatment was in time for her. There's still a lot of hope. But the reason I'm sitting here today is not because of my daughter. And so I think that I can tell you that myself, Dr. Yu, everyone at NCATS, so many people at the FDA are really aware that there is, at least I can only speak for Mila's type of treatment right now, is that this is a potential platform that cuts across many diseases. This is not about Mila, and it's not about Batten disease. And I think people are really motivated to twist their head around 180 degrees and think outside the box and really try to push this forward. And I like to use the word aggressively together with safely, because children are dying. And I hear about it every day and every week. And I see pictures and I receive emails. And I stay up at night thinking that a treatment like Mila's could help, call it 10% of cases of monogenic diseases. There are many more other tools that are being explored right now, and that I see finally, after 3 years, I see real commitment from kind of every player in this field to really try to aggressively, safely try to make this happen across the board. So I can just tell you that we're all here because we're really working really hard behind the scenes to make this happen, and I think that we're going to see change. I don't know if it's going to be 100 kids in the next 3 years, 1,000, 1, I don't know, but we're working hard to make that happen. And I'm trying to be the voice for the many people, parents out there where the stars may not have been able to be aligned as much as they were for Mila.
Thank you for being a warrior for us all.
Thank you.
And I would say that seems like a perfect place to end. So I want to thank all the panelists for their participation. Thank you all.